So, this is actually the last lecture of this week, which is actually a review lecture for what has been, whatever material has been covered so far in the first 3 lectures. So, we introduced in the first lecture the subject of biomems and microfluidics. We talked about the various definitions. Biomems is defined as a biological or biomedical application of MEMS or micro electromechanical systems technology. We also talked about bio-nanotechnology, which is actually the biological applications of nanotechnology, better known as science of objects which are smaller than about 100 nanometers or so. We briefly described microfluidics as a study of fluid transport at the microscopic length scale, and we also talked about the various utilities that such systems would have. We started with the scale and size concept, which would give you an idea of the emergence of the field and why it is needed, wherein we discussed various size issues related to the biological species. We talked about bacterial cells, which are about close to more than a micron size. We also described viruses, which are a few hundred nanometers, starting up from molecules. Describe the one helical turn of a DNA to be about 2 to 3 nanometers, 20-30 angstroms, and we further emerged into the highest size cell, which is most plant and animal cells, which are more than 10 micron sizes. While comparing them with the objects and systems which are possibly fabricated using MEMS technology, which could size very well or rhyme very well with the sizes of these biological entities. We talked about these scale issues as a basic reason for the emergence of these 2 fields, where one field would concern the top down approach of fabrication from a higher size wafer to various smaller elements of the sizes of these entities, and the other side would be a bottom up approach that Mother Nature typically has from the smallest elementary atom or molecules all the way to the most complex form. And then we said that because these 2 areas are synergistic, there is a scope of match of the dimensions between both the areas. Therefore, automatically a field of study called biomems emerges, where the detection of the entities, biological entities, can be very easily done with some of the sizes that miniaturization has to offer, mass or of sizes equal to that of the biological entities. We further defined microfluidic transport and talked about Reynolds number. We also discussed about the implication of Reynolds number on the laminarity of the flow. The example that was given was the mixing or the parallel flow of 7 different dyes in a small channel, where the dyes would emerge as such without any mixing if there would be no diffusive transport. Also discussed was the benefits that microfluidics would provide by giving a small volume of fluid which needs to be thermally, which needs to be in various applications thermally cycled, and the advantage that microscale would provide is a very high heat transfer coefficient in such small volumes. Also we discussed about various aspects of mixing and how Reynolds number, low Reynolds number promotes this concept of band like flows, which would not promote by and large mixing. And then we also discussed some problem examples from the actual MEMS domain, where we would describe the application of a microneedle or even the applications of MOS transistors towards single cell and the way that the cells would behave with respect to each other in unison while sitting over an array of MOSFETs. We also described the micro cantilever array and how it could be used for biological detection, and also talked about the concept of lab on chip, where a series of biomolecular reactions from the laboratory could be carried out in a small chip level of less than an inch by inch size. We further analyzed different real life MEMS applications, more concerned with physical MEMS. where we would talk about things like single chip accelerometer or single chip microphone, and also describe about the digital micromirror device. We also mentioned about how MEMS technology is useful for fabrication of micro cantilevers, and then we started with sensors. Okay, and we actually related to develop a sensor model which would comprise typically of a recognition element, a transduction mechanism, and generate a signal which would be more machine readable. In fact, the sensor was laid out as something which would analyze from variety of analytes selectively one particular analyte, and that would happen with respect to a recognition element which would be able to recognize that particular analyte. This recognition element would typically be mobilized on the surface of the transduction assembly or the transducer, which would convert the chemical signal or whatever signal comes from the analyte into a machine readable signal, which could be an optical signal or an electrical signal, and the signal would be transmitted to the processor, which in case of a fully automated system would be the brain of a computer, and in case you talk about human systems or biological systems, it could be the brain of a human being or a living being. Okay. We described sensors and classified them into three categories: physical sensor, which would typically look into all physical parameters like measurement of properties like temperature, distance, pH, temperature, pressure, mass, so on and so forth. We also talked about chemical sensors, which would measure typically the chemical substances or chemical responses selectively. We further describe biosensors, which typically would use a biological recognition element and would be responsible for detecting a biological molecule or a biological species. Further, sensors were the sensor model was outlaid for a variety of different situations. Like one was the litmus paper. We described the litmus paper as having a content which would be the dye of the litmus paper, which would change the coloration to be the recognition element, which would be sensitive to the pH or hydrogen ions in the solution. And then further, the transduction that would take place would be in terms of a change in the absorption spectra or a color change of that particular dye molecule, finally resulting in giving a visual display of such a pH change in terms of a color change. We further also explored different organs that human beings have, the sense organs, as sensor models. For example, we talked about the human nose, and we mentioned that how a series of chemical reactions which would lead to a sort of electron release would take place at the cilia of the olfactory cells, which is also in the region of the mucous membrane. It's a part of the mucous membrane, and basically what it would do is it would try to synthesize a series of proteins at a very high rate, which would interact with different constituents of the filtered air which would go into the human nose system, and thus result in some electrochemical changes, and this would eventually lead to a readout of smells like, and readout and classification of smells like sweaty, pungent, sweet, rotten, so on and so forth, by means of different rates of electron release, and this finally would go up into the brain through the olfactory route, so that you can distinguish between these smells, and the defense mechanism of the body can then generate some actions, okay, which would avoid those smells which are particularly pungent or sweaty, etc. We also described about the human eye, and we talked about the retinal, the retina, which is actually at the back of the eye, and it comprises of these rhodopsin molecule, which is an opsin protein, and typically it is linked covalently to a compound called retinal, and there are two isomers of retinal, one is a cis and a trans isomer, and what typically light would do is to fall on this part of the rhodopsin molecule and change the cis to the trans state, thus resulting in again an electron release, and therefore different intensities of light like bright light or dim light could be categorized suitably by means of such electron releases, which would then flow through the optical nerve all the way to the brain. So that is how we categorize the various sensor models in the biological systems. We further described the various aspects of the sensor design, like recognition elements, transduction types, method of immobilization, performance factors of the sensors, so on and so forth, and finally studied these independent domains one by one. For example, we mentioned about ion selective electrodes in the recognition element, and there typically the selectivity of a recognition element would really introduce the sensor to respond to one analyte from a group of different analytes, okay, and it would also try to avoid interference from the other analytes which are present in that sensing modality. In that context, we discussed biosensors particularly, which would be using recognition elements like antibodies or enzymes or even nucleic acids and receptors, which would typically identify one particular species. Here in the example that was given was that of glucose sensor, where the enzyme GOD or glucose oxidase would be able to analyze only or catalyze only the glucose molecule into gluconic acid and H2O2, thereby increasing the pH of the medium. So basically the selectivity component sometimes comes from the recognition element, sometimes it comes from the design of the electrode which is sensing the particular analyte of interest. In this context, we described the enzyme linked immunosorbent assay, ELISA, which talks about the detection of HIV species within the infected patients. And the idea which is projected here is that there is a glass plate in which we coat the antigens of the HIV antigens particularly, and the logic that is used is that if this HIV species are present within the human system, it causes the defense mechanism of the human system to generate antibodies, and these antibodies are so called anti-HIV antibodies, which would be binding in nature to the HIV antigens. So if there is a coating of the HIV antigens on the plate, the glass plate externally, which is done in vitro, and the patient's sample, the blood sample from an infected patient or otherwise healthy patient is put separately on these coated plates, the requirement of the antibody being in the blood sample would allow the antibodies of an infected patient's blood sample to bind to this HIV coating which is there on the top of the glass surface. And for the otherwise healthy patient, because the antibodies are generally not produced within the body, there is no presence of any HIV antigen within the body, there won't be any binding event there. So what is practiced at the third stage is that there is another antibody, a secondary antibody, which has an enzyme typically, which would create a change in color of a chromogen or a color dye. So this antibody capable of binding to the anti-HIV antibody is basically led in the plate. Now the plate has a stack now, there is an antigenic layer, there is the human anti-HIV antibody generated from the human system. We wash off everything else which is there, and then there is a secondary antibody which is coming as a bundle, as a stack layer on the top of this primary antibody and binding to it, and the secondary antibody has the enzyme. So if the patient's sample, the patient's blood had the antibody corresponding to HIV, then this whole binding event and this 3 layer would generate, and there would be the presence of the enzyme. So when there is a fourth step where there is a chromogen or a color dye which is put inside the system, immediately there would be a change of color and change of the absorption spectra. So the quantitation of the degree of change of the spectra would also define the amount of antibodies which are present in the patient's sample, which is a very good indicator of the extent to which HIV has infected the patient. In the other case, of course, for the healthy patient, because not even the primary antibodies for corresponding to the anti-HIV was there in the blood sample, there won't be any such stacked binding, and there won't be the presence of enzyme, because we have a wash step in between every event, and if there is no covalent linkage, whatever is on the plate would typically be carried away. So you would not have any color change in that particular case. We also described about various other modalities related to sensor design, like transducer development, where we specifically mentioned that most of the transduction which is available today, particularly on a commercial scale, is electrochemical, and the reasons that were indicated were that electrochemical transduction is always more amenable to reading out devices because they produce electrons as a result of their sensing modalities, and thus it is more integrable with microelectronics, or it is more integrable with processors, okay, which makes this mode of sensing very useful because you can quickly process the data coming out of such a sensing method or a sensing modality. We also described about the new thrust area of photon driven devices, where there is a change in terms of the number of photons generated. The transduction is having a final signal in terms of a light signal with photons, and there is an increasing thirst because of the rapidity of the photon generation and process of transmitting the photons. The overall rapidity of the sensing system should also increase, and therefore there is a tendency of increasing to shift towards this photon driven devices, although they are far away from commercialization as on date. We also talked about mass based sensing, where small change in mass of the micro or even sometimes the nano level can be very sensitively detected by means of cantilever devices and sometimes piezoelectric crystals. We talked about different transducers which would produce a change in the electron flow. Herein we mentioned of course different electrochemical sensing modalities like potentiometry and voltammetry. We also described conductometry and impedometry as some of the futuristic modalities of sensing. We also talked about the various methods of immobilization, mentioning herein that the recognition element which we need to finally immobilize on the surface of the transducer for a sensor needs to be put in place, and there are various ways of doing that immobilization. There could be a covalent attachment of the species to the surface, there could be a attachment through a ligand, there can be a ligand chemistry, a linker molecule between the sensor surface on another side and the recognition molecule on another side. There would also be a method of microencapsulation where we typically trap these analytes between two or more membranes. There could be a physical entrapment using something like a gel or a paste on the surface, and then trying to immobilize the recognition element within that gel at various levels. Each of them have their own limitations, and some of them also in terms of advantages in terms of fabrication process can be classified into various types. We described performance factors where we would look into the different yardsticks available for a sensor engineer to distinguish the efficacy of the sensing device. For example, one such parameter could be the selectivity, the ability to distinguish between different analytes of interest. Another could be the range of sensitivity, up to what millimolar level or micromolar level of a change of concentration can the sensor distinguish. There could also be a question of accuracy. What is the percentage within which the sensing domain would operate accurately, giving replicable readouts every time. And then we talked about various other performance yardsticks like response time, recovery time, working lifetime, so on and so forth. Then we shifted on to description of biochips and lab on chips. We briefly talked about the very wide presence of lab on chip devices in the area of integrated gene analysis and detection, and we also described some novel tools of nanobiology, where again how a single cell behaves in terms of producing molecules, thousands of molecules on its surface, could be very useful information to know about the collective or individual behavior of the cell, and this data would be of immense use to the physiologists. And this whole new area of nanobiology was supposed to generate those tools, particularly with the integration with the micro domain or the nano domain, integration with the electronics, that such tools would give you a very rapid sensing mechanism for understanding how the molecules start producing on the surface of a cell, for example, or how they start disappearing. And individual cell to cell communication would then be deciphered in terms of electrical impulses or responses. Herein we also mentioned about the processes of transcription and translation, where DNA would typically be coded into a messenger RNA, a much shorter transcript of the DNA, and then it would eventually be translated into altogether different language from the sequence of nucleotides to the sequence of amino acids. Okay, so the final goal would be to produce the sequence of amino acids which are otherwise known as proteins, and this mechanism would happen within the whole cell. We further started with some introductory topics in the area of microfabrication. We described about surface and bulk micromachining, and we also talked about the various materials which are used for the purpose of making the devices. There were 3 different generations of such materials. There would be silicon, glass, of course, because most of the MEMS processes are borrowed from the microelectronic industry. There would also be polymers which would be able to typically rhyme very well or behave very well in presence of the biological species. And then of course there would be a completely new class of MEMS devices which are made up of the biological entities themselves, like let us look at, for example, DNA printers or even cell based devices, so on and so forth. We described different processes where, with a series of etching steps and selective lithography steps, we could make small crevices and vias with thin membranes standalone on the top of them. And we described how to make briefly such a membrane, either it is of a beam like orientation or a cantilever like orientation. We started off with some description of concepts like what etching would be at selectivity, and then of course we just started introducing the photolithography process and photoresist. So this sort of concludes what we have learned in the first week of our lectures. I would eventually also like to provide an assignment at the end of this lecture, which you have to submit back to us. Thank you.